Good morning. This is Christiane Pixel. I'm with Pelo Biotech, and I welcome you to a new episode of Pelo Academy. Today, we have a special guest. This is Tommaso. He's here from Italy, and we named this webinar Breast Cancer, New Perspectives in Vitro Studies. Well, that's funny, huh? We always tried so many times. And breast cancer is considered one of the most prevalent cancer worldwide, Tommaso, and I'm very looking forward to your lecture. Yeah, so Please go ahead. I will turn off my camera as well.
Thank you, Christian. So good morning. I'm Tommaso Sbrana, and I'm the CEO of IVTech. Today, I will introduce scientific data produced by our clients in the field of in vitro studies to evaluate the breast cancer scenario. Before to start, I would like to introduce a little bit about myself. I'm a biomedical engineer. In the last 14 years, I focused my attention on the development of new tools to increase the predictivity of in vitro models. In particular, at the beginning of this period, I focused my attention on, when I was a PhD student, on the design and characterization of these new tools to overcome the limitation of multiple plate and transport system. Then, in 2014, in collaboration with some of my colleagues, I founded IVTech. So I shifted my focus from the research to the business related with the commercialization of these products. So, as we know, breast cancer is one of the most prevalent cancer worldwide. In 2020, it has affected up to 2.3 million women. We recognize different subtypes of this pathology, but for sure, the most prevalent and aggressive is the HER2 enriched subtype. So in this presentation, I will introduce three different in vitro models. The first two models will be focused on this particular scenario. So this pathology is related with epidermal growth factor receptor called HER2, and this receptor is in correlation with the growth of the cells. In particular, the overexpression of this growth factor is related with the overgrowth of the cancer cells. It is possible to treat this pathology with a drug in order to reduce this, the proliferation of these cells. And one of the drugs that can be used is trastuzumab. However, it seems that a significant number of patients that is HER2 overexpressing breast cancer positive show resistance to this drug. So the researcher at Fondazione Pisana per la Scienza, a famous research institute in Pisa, Italy, has studied this scenario using an advanced in vitro model. So this was the platform that has been used. It is provided by IVTech, but I will introduce the technical details in the second part of the presentation. However, the platform is composed by a Pristility Pump called the LiveFlow, cell culture chamber called the LiveBox1, where we are going to place the cells. We have a fluidic circuit in order to let the medium flow in this setup in order to expose the cells to a dynamic environment. So the first model is based on MCF7 cells. They have cultivated the cells following a 3D on top methodology. So basically, they coated the glass disc in the LiveBox1 using Matrigel, then they plated the cells, and again Matrigel on top. After a few days, they obtain a cell aggregate in a sphere-like shape. After 24 hours, they started the dynamic stimulation. In 3 days, they have the first endpoint analysis for the 2D static control, and after 6 days, they conclude also the dynamic stimulation with the endpoint analysis. From the immunochemistry analysis, it is evident that the cell aggregation in a sphere-like shape after 5 days. This spheroid reached up to 200 micron in diameter after 6 days of dynamic stimulation. In correlation with the development of this 3D structure, we also have the development of hypoxia domain, as it is demonstrated by the green color in the picture in the slides. This analysis can be performed using an inverted microscope or an advanced optical imaging platform in real time. Administering the trastuzumab permit to evaluate the cell viability. So after 3 days, we have a relevant, significant reduction of cell viability if we compare the 3D configuration with the 2D configuration. And in particular, it seems that in 3D configuration, we have an increase of resistance, of cell resistance, if compared to the 2D configuration. The other model that I would like to introduce you is has been studied always at Fondazione Pisana per la Scienza, and it is based on the use of BT474 cells. In this case, the researchers cultivated these cells in a dry alginate sponge. After a few days, again, they obtain a sphere-like model. In 24 hours, they started the dynamic stimulation, administering the trastuzumab at 50 micrograms per ml. And after 3 days, they had the first endpoint analysis for the 2D and static control. And after 6 days, they conclude the 3D dynamic stimulation. From the immunochemistry analysis, it is evident the sphere-like shape, as demonstrated by the section reported in the pictures. Administering the trastuzumab, we have a reduction of cell viability into deconfiguration, as it is expected, but it's even more interesting to analyze what happened in the 3D scenario. So on the top graph, we have the control in static condition, and on the bottom side, we have the dynamic condition. As you can see, after 6 days, we have a significant reduction in cell viability if we compare the treated and not treated configuration. So it seems that in a 3D and dynamic condition, we are more sensitive to a drug such as the trastuzumab. This result has been also confirmed by the gene expression analysis. And in particular, in the histogram on the top, you have the cyclin A expression, and on the bottom side, the cyclin B1. So in both cases, we have a significant reduction of the gene of this gene expression in dynamic configuration. In conclusion, it is possible to replicate complex scenarios using an advanced in vitro model. In particular, it is possible to study the cell resistance that is shown in 3D configuration if we compare with the 2D configuration. In dynamic condition, it seems that we are also more sensitive than in static configuration. The dynamicity allows also the development of hypoxia domain. A different procedure, a different approach has been developed at the University of Pavia. In this case, researchers analyzed the tumor signaling, focusing the attention on the metastasis model. So they decided to develop a target tissue, a healthy tissue in a 3D configuration, and then they injected this metastasis model in the fluidic circuit to evaluate the interaction mechanism between this circulating metastasis and the healthy tissue. So the first point was to develop the healthy tissue, so the target tissue. They used fibroblasts cultivated on a polystyrene porous scaffold. In 10 days, they started the dynamic stimulation, and the day after, they injected the metastasis model. At day number 12, they had the endpoint analysis. So let's focus the attention on the target organ, fibroblast. This is a a preliminary model that will be substituted by a bone model in the further experiments. However, fibroblasts are available and proliferate during the whole experiment time frame. And these results is confirmed by the increase of fibronectin production, as you can see in this table. Focusing the attention on SEM analysis, it is possible to evaluate the fiber of the scaffold, and between these fibers we have cells elongation. These are our fibroblasts. So the healthy tissue is ready to interact with a metastasis model. So let's analyze the metastasis model. The researchers started from MCF7 cells, so breast cancer cell line, and lung cancer cell line. They cultivated these cells on a polyhema coated plate for 21 days in a DMEM and neurobasal medium in a ratio 1:1. After this period, cells start to grow in suspension and duplicate. The spheroid that they obtain has been gently dissociated and loaded with calcine in order to evaluate in real time their presence in the fluidic circuit. So, as I told you, once this metastasis model was ready, they injected in the fluidic circuit. The researcher has characterized this metastasis model with gene expression analysis and protein analysis. I will not talk about this data, but if you're interested in, you have the reference to the paper in the bottom part of the slide. So, once they injected the metastasis, these cells interact with the healthy tissue, as demonstrated by the green color in the picture in this slide. So at this point, the question is, are the metastases interacting with the fibroblast or with the scaffold fibers? So in order to answer to the question, they performed a SEM analysis. And as you can see, this is a nice metastasis that interacts directly with the fibroblast fiber. So the interaction between the metastasis and the fibroblast is not scaffold mediated. So at this point, the in vitro platform is ready to be used to test, for instance, the effect of a drug. And in this case, the researchers analyzed the effect of 3 different integrin antagonists in order to reduce the adhesive mechanism, the adhesive properties of the metastasis onto the target tissue. They evaluated 3 different compounds, and only silingitide, both at 20 micromolars as well as at 50 micromolars in concentration, demonstrate a significant reduction in metastasis adhesion with fibroblasts. So this could be a good candidate for further analysis in this field. So in conclusion, using a Livebox 1, it is possible to maintain high proliferation of 3D healthy tissue. It is also possible to mimic the interaction between a circulating material and this 3D tissue. And also, it is possible to evaluate the effect of a drug such as the silingitide in order to, in this case, reduce the effect, the adhesive properties of the metastasis onto the healthy tissue. So at this point, I hope I get your attention, and I would like to introduce you IVTech and the technology that we offer. So IVTech has been founded in 2014 as a spin-off of the University of Pisa. In the next year, in the following year, we have released several products in order to increase our products portfolio. In 2021, we had up to 7 chambers and 3 Pristility pumps. And in the meanwhile, we have signed an agreement with different distributors in order to distribute our products in Europe and outside European borders. Our goal is to become a reference partner to develop advanced in vitro models. Our core values are related with innovation and professional approach to increase the predictivity of human reality. But we want to maintain a versatile and sustainable approach from an economical point of view. Our business model is structured on three main pillars. The first one is to provide the technology through a distributor network. The second one is to help our customer developing engineering services related with the design of these advanced in vitro models. An example of this service could be the evaluation of the parameters to simulate the in vivo scenario, or even a mathematical model to downscale the dimension from the reality to the in vitro model. The third pillar is represented by the possibility, the opportunity to develop also the biological aspect related with these advanced in vitro models in collaboration with certain biological partners. Our mission is to fill the lack between a 3D and static model and a 3D and dynamic model. But this is not sufficient in order to replicate the human reality, because as we know, the response to an exogenous stimulus is the results of a cross-modulation between different organs, different tissues. So we want to add this complexity to our in vitro model. So for us, advanced in vitro models is represented by a 5D in vitro model, 5 degrees of complexity. The third three degrees are related with the topology, the three-dimensional topology of the model. The fourth degree is related with the dynamicity of the environment, and the fifth degree is related with the correlation, the cross signaling between the tissues. So this is an overview of our technology. As you can see, our platform, our fluidic platform, is a very compact product. It is composed by a peristaltic pump, live flow, one or more chambers called the live boxes, and a fluidic circuit where we have the flow of medium which recreates the blood action in human circulation. The field of application is very wide. We have studies in metabolism, test of new materials, but also simulation of disease scenario. So the positive tick is related with published data, but we have also ongoing projects. In detail, we offer Livebox 1 is a single flow bioreactor. I already mentioned in the study that I described you before. It is developed for attached cell culture condition. We can work with both cell line, primary cells, or even biopsy. It is equipped with a removable glass disc on the bottom side and on the top side in order to recollect all the biological material at the end of the experiment and perform a post-processing in a Petri dish or in a plate. Livebox 2 is a double flow bioreactor equipped with a removable and porous membrane. It has been designed to work with physiological or pathological barriers, both in air-liquid or liquid-liquid interface. We have also our live flow, which is our peristaltic pump. It's a very compact product compatible with the incubator environment. It's equipped with 2 removable and autoclavable heads, driven by 2 independent motors. So we can work with 4 fluidic circuits at the same velocity, or we can modulate the velocities in order to increase the complexity of our model. For instance, we could have a barrier, an intestinal barrier, where we will set up a flow rate, a certain flow rate on the top level, the lumen, and a different flow rate in the shell in order to mimic the physiology. It is possible to use a standard cell culture protocol in our products because of the compatibility in cell growth area with the standard slot of multi-well plate or even transwell. The protocols, the procedures that we have to follow are very similar to the standard protocols that we are used to follow in a standard multiple plate system. So as you can see, starting from a sterile chamber, we have just to open the chamber. We have to seat the cell suspension. In case of a floating scaffold, we can fix in position using this plastic disc. We close the chamber and we clamp the system in order to assure the water tightness. So tissue or cells will mature in an incubator environment. Once the tissue will be ready, we will start the dynamic stimulation, joining the chamber to a fluidic circuit and a peristaltic pump. It is possible also to follow standard procedures to monitor the cells. So the chambers are transparent in Z-axis, so we can monitor in real time using an inverted microscope or even an advanced optical imaging platform. As I told you, the glass discs are removable in order to recollect the biological material as well as the membrane in case of live box 2. The chambers are modular, so we can join together different modules, each one focus on a different tissue. This is the so-called multi-organ approach. So we have different tissues which communicate through an exchange of liquid. And this is crucial once you have to test the effect of a drug in order to test the principal effect onto the target organ as well as the related effect, the side effect onto the related tissues. For us, it's also important to be able to evaluate, to mimic the pathological environmental condition, because what I described to you is suitable for a physiological in vitro model, but if we want to evaluate a disease, we need something else. So we provide our live PA, which is a pressure modulator, and it's used to increase the pressure inside of our chambers, so onto a tissue. This has been used to test, for instance, the pathological condition related with hypertension or glaucoma. It can be used also to evaluate the scenario of glioblastoma, so the presence of tumor mass on our healthy tissue. So in conclusion, IVTech products allow to use the standard cell culture and monitoring protocols. It is possible to recollect the biological material at the end of the experiment. The whole platform is compatible with the incubator environment. Cell culture chambers as well as the fluidic circuit are autoclavable and reusable up to 10 cycles of autoclave. It is possible to join together different modules, different tissues in order to develop a multi-organ approach. And it's possible also to recreate the disease model related with an increase of pressure using our live PA. So that's all for this presentation. You have here my email, and if you're interested, please follow us on LinkedIn and on our YouTube channel. Thank you for your attention.
Yeah, thank you, Tommaso, for the great presentation. Also, if you have any questions, you can also go through Pela Biotech. We already know a lot about IVTech, as we are partners for a very long time now. I think it's almost 6 years or so, Tommaso, yes. Right?
Yes, exactly.
Yeah. And yeah, thank you for that. So if you have any questions, just let me know right now. You can type those in, and I hope then that we can answer them. If not, you can always ask later on. But I realized, Tommaso, that you have a new logo, and it says "Closer to Humans." This is very nice.
It's right.
Why did you change it?
Well, when we started with our company, we were mainly focused on the technology. So for us, IVTech was a company to provide technology to the clients. But in the last years, we increased our offer with services, and in particular with biological service, in order to develop, for instance, a skin model based on human biopsy in order to test, for instance, cosmetic compounds. So we changes our tag with closer to humans, because for us it means that it can cover also this aspect. For the logo, it seems to be related with cells. It could be a cell that moves in order to develop in a 3D configuration, or with the dynamicity which characterize our in vitro environments.
Yeah, that's nice. Okay, so first question would be, Gloria is asking, is the system suitable also for organoids?
Yeah, it is. In case of organoids that need floating condition, we can use Lightbox 2 in the so-called sigmoidal flow configuration in order to maintain an organoid or a spheroid or a cell aggregate in floating condition in a bottom chamber. So this biological material will not flow outside of the chamber, because the flow will pass through the chamber in a vertical direction, and the membrane will confine the biological material in the bottom environment of the Lightbox 2.
Okay, yeah, thank you for this. I hope this answered your question. If not, she said thanks. So yes, it did answer the question. I also saw, Tommaso, that you can use the chamber for multi-organ testing. How many organs can you test in a row?
Well, the connection of different chambers in series increase the complexity of the in vitro model. So we are able to connect up to 10 chambers, but currently our clients are reached up to 4 chambers, so 4 different tissues connected in series. You have to consider that one thing is the feasibility, and one thing is the possibility to control this complex environment. So between 4 and 5 tissues, it's okay. We can control in an easy manner.
Oh, that's. Have you ever tried with 10, or somebody else to try with 10 chambers?
We have tried from an engineering point of view to interconnect 10 different chambers in series, and we obtained good results. But as I told you, once we have the biological material, we need to control, to monitor each module. So having 10 different modules in connection, the complexity increased dramatically.
Yeah. What I really like about your system is that it's such an easy setup, and you can also use this at the university, because it's so easy to use. And there's one more question. Let me check. How long does this take? And how exactly do I have to set it up in advance?
Well, as I show you, the protocols, the procedures that are required to work with these products are very close to the standard cultural protocols or monitoring protocols. So we have experience to train our our clients during our workshop. So in general, we take let's say 1 day of training in order to let the researcher perform by himself or herself the experiment. So we also have video protocols on our YouTube channel. So we promote a closed talk between the company and the customer in order to go faster in the first phase. But however, I would say that in a very short period, researchers are able to perform the first test, the first experiment by themselves.
Okay, great. There's another question, Tommaso. Is a chamber in which you grow the cells in plastic? Maybe you can go a little bit further where you showed the setup.
Well, in plastic, you mean the substrate where we are going to plate to see the cells?
Yes. Yeah.
Okay.
She said yes. Thank you.
The disc which represents the basal surface, the bottom surface of both Lightbox 1 as well as Lightbox 2 is a glass disc. But on the market, we also have the same product in a plastic material. So it's easy to change this removable slide with a plastic slide.
Okay, I hope this answered your question. Okay, very good. Okay, so if you have any more questions, just let me know, and Tommaso is very happy to answer them.
Yeah.
I think you already published a white paper on this topic, not only the presentation. So if somebody is interested in, maybe we can send this over. Just give us your email address. I think this would be fine, Tommaso, right?
Yes, of course.
Yeah, thank you. Okay, and yeah, if you have any more questions, just type them in. Now is the time. And I also like to let you know that we are growing. Our team is growing very much here at Pelo Biotech, and we are looking for 2 new Pelo family members, and one for the logistics department, and one would be working with me and Peter at the marketing. So if you know somebody who knows a little bit of German and English, just let us know, and we are happy to work with you. Also, I like you to join us on our social media channels. We are on Insta or Meta, whatever, and Twitter and LinkedIn. Also, we have some things at ResearchGate, so I would be very happy if you would join us there, and also go through our website if you have any more questions. And yeah, stay tuned in for our next webinars. We will send you the recording afterwards so that you can listen to it. And also we will publish this on our YouTube channel and on Tommaso's YouTube channel. Okay, Tommaso. So I hope we are very much looking forward to it. I think we will also have a next webinar in the upcoming weeks. We will schedule this. And this will be also a very exciting topic. If you would like to know more about this technology or on a specific topic, also let us know. Tommaso will be very happy to talk to you or do even a webinar about it. We can also do a mini webinar for your working group, yeah, for your colleagues, and to help with the setup and everything. And so I just let you go now and hope you have a wonderful day. Thank you, everybody, for joining. Please stay tuned in. And as I told you, you will find the recording of this webinar on our website. This is www.pelobiotech.com. Sorry about this, I was just reading. Thank you, Gloria, and have a wonderful day, everybody. Please share this information with your friends and your coworkers, and talk to you soon. Bye.
Thank you very much for your attention. Have a nice day. Bye-bye.
Thank you, Tommaso. Thank you. Bye-bye.